Today in this video we are going to study about what is photorespiration, why it creates a big difference between C3 and C4 plants. In our last classes we have already discussed what are C3 and C4 plants. C3 and C4 plants are basically those plants which have 3 carbon and 4 carbon as 4 carbon molecule as their 4 carbon atom molecule as their first product. The first step of photosynthesis, or you can say Kelvin cycle, in normal cases, that is when carboxylations occur. In the C3 pathway, the enzyme ribosco catalyze the first step. Ribosco combines with RUBP, that is ribulose 1,5-bisphosphate, with CO2, forming two molecules of 3-GPA, that is 3-phosphoglyceric acid, and this is productive, which leads to the formation of sugar, right? But ribosco is a dual nature enzyme. What do you mean by that? Ribosco acts as both carboxylase and oxygenase. Something about ribosco. Ribosco is one of the most abundant enzyme on the earth. It has active sites that can combine with CO2 and O2. It has competitive binding. Competitive binding means if the amount of one exceed over other, that molecule which is excited will combine with the ribosco. Here it is explained better. Which one binds depends upon the relative concentration of CO2 and O2 in the leaf. If CO2 binds, Kelvin cycle will proceed, and if O2 binds, a different cycle or reaction will proceed, known as photorespiration. Photorespiration in C3 plants. Why in C3 plants and why not in C4 plants? We will discuss this further. Sometimes O2 binds with the ribosco instead of CO2. This then ribosco RUBP react with oxygen and produce one molecule of 3-PGA, phosphoglyceric acid, and one molecule of phosphoglycolate. Glycolate is a 2-carbon compound which enters the photorespiratory pathway. Why it is called loss process? Because no sugar is formed. ATP or NADPH is produced. Instead, the ATP is consumed and CO2 is released. That is why it is called a wasteful process. Why now here's the question: why C2 cycle, or you can say photorespiration occurs in only C3 plants, not in C4 plants. Here are some reasons. C4 plants have CO2 concentrating mechanism. Remember, we have learnt about OAA, oxaloacetic acid, right? It is a 4-carbon molecule which later converts into malate or aspartate. These acids moves to bundle sheath cell where they release CO2. This increase in CO2 concentration around ribosco. Ribosco is mostly carboxylase, not oxygenase. Why? Because there is availability of carbon in the form of malate or aspartate. Photorespiration is then suppressed because of availability of carbon dioxide. That is why in C4 plant have higher productivity or yield. They can tolerate higher temperature better than C3 plant. Here is a visual representation how C3 and C4 plants act on unavailability of carbon dioxide. In C3 plant, RUBP combines with O2 to produce two types of molecules. One is phosphoglyceric acid and one is phosphoglycolate. It have 3 carbons and it have 2 carbon. This is completely waste because it cannot undergo or participate in Kelvin cycle, whereas the second molecule it can participate in photosynthesis at the mid of Kelvin cycle. But in C4 plant, due to the availability of this carbon dioxide, it combines with RUBP and form two molecules of PGA, which inhibits the process of photorespiration.